expertise to develop a lot of computational omics. So she deals with cancer genomics, metagenomics, and comparative genomics. And with all this research that's happening in antimicrobial resistance, there are chemists collecting antibiotic data, microbiologists collecting all these antibiotic resistance genes, and engineers doing all the other aspects of environmental pollution. We have all this vast data that can only be made sense if we have a bioinformatics expert. So I have a list here of all the things she has developed, but I think I'm just going to have her talk about that instead of me telling you what she has developed.
Thanks, Diana, for inviting me to come here for this wonderful workshop. I met some of you guys. We had very inspiring conversations about the AMR. I'm sure you will see or hear a lot of AMR related work today. Thanks, of course, for all the hard work of the organizers. This is I'm very excited about this discussion and finding out more in this area of antibiotic resistance. So my background, as Diana said, it's very diverse. So I trained initially molecular evolution, population genetics, and then I even really worked with plants, humans, and then now my really recent research interest or thrust is all about antimicrobial resistance, developing computational resources for mining microbiome data. So let's look at, so I'm in the computer science department at Virginia Tech. So here's my talk outline. So I will briefly introduce you to metagenomics and how it's applied to studying antibiotic resistance. So it's going to be a switch of kind of mode. Everybody else before me already told you a lot about antibiotic resistance. So I will have some similar slides, but they made the introduction to save me time. So I will be focusing on mostly the computational resources we developed for this area, mainly focus on the systems that we have in place: Metastorm, Deep ARG, ARG minor, Nano ARG, and Metacompare. So okay, what is metagenomics, right? So here's the slide on shotgun sequencing of metagenomics. So the idea here is that you can look at anything you are interested. So you can dig up some dirt, or you can go to the ocean, or Arctic glacier, or you can go to this is the human gut. So you can take some samples from there, extract the DNAs, and then sequencing them. So the sequencers will generate lots of sequences, DNA sequences, ATCGs, right? So in short ones, so lots of them, hundreds of millions of them. So it's big data. So easily the data regularly we're handling 100 gigabytes of data or terabytes of data. So from here, once you generate these DNA sequences, okay, through the sequencer, it looks like this string of ATCGs, and then it goes through the computational pipeline. So to annotate them. So annotating them is, so you have these small segments. So you can annotate them to answer mainly three questions. So the first one is looking at these environmental, different places. Your question would be, who is there, right? What kind of bacteria or microbes or viruses are there? So you want to look at their abundance, the diversity of the bacteria or viruses, or you want to look at how abundant they are, the community of members. The second question you want to answer is, what are they doing there? So basically, you want to look at the functions, okay, what kind of biological functions they may have. The third question is, why are they there? So this is more looking at ecological relationship between the members of the community. So these are three major big questions people would like to answer or use metagenomics to hopefully answer these questions. Okay, so we talk about metagenomics. So how do we analyze this big data? Right? So we have lots of ATCG sequences, and how do you analyze them? So here, this is like a cartoon illustration of the process. So you have linear sample from various different places. So there are microbes. Then going through the sequencing machine, you have the sequences, DNA sequences. Then there are two branches from here. So one is reconstruct the genomes, right? So these are random pieces from the organism. Okay, so you can try to put them together. That's so-called assembly-based mechanism. Try to recover the entire genome. So that's from the recovered genomes. You try to guess what kind of bacteria are there. Okay. The other route is really through so-called marker gene database comparison. So here again, these DNA sequences, you have lots of them, and you compare them with a reference database, so-called reference database. Then based on the sequence similarity between the collection, the random pieces of DNA sequences with the reference databases, you're trying to guess what these pieces, where they come from, or what kind of genes they might be, etc. So it's based on sequence. The bottom line is based on sequence similarities. So here, this is called direct annotation. You can identify, things like what kind of genes are in your sample, and you can also count the abundance of those genes. Okay. So depending on, I will introduce its link or application to antibiotic resistance, studying antibiotic resistance genes. So here's the antibiotics resistance, how it's developed, and this is an example of how antibiotic resistance spreads. So my colleagues already introduced different pictures. They are all taken, from different resources and internet or literature. So without saying antibiotic resistance is real, the threat is real and it is serious. So the thing here is really by 2050, this I remember Corey showed something slightly different, a different picture. But the point is here, by prediction, by 2050, the AMR, antibiotic microbial resistance, is going to overtake the cancer, killing people in terms of the death caused by antibiotic resistance. So it is real. We need to act urgently. Okay, so I talked about metagenomics and how can it help to study antibiotic resistance. So here's again that figure that I showed you. So one example here, if you want to look at, so you generate DNA sequences, random pieces from the environment, and then you want to look at in your specific area, in terms of antibiotic resistance, you can compare, for example, this database. This database can be a set of antibiotic resistance genes. So by comparing these random pieces to this reference database of antibiotic resistance genes, you can kind of guess or infer what kind of antibiotic resistance genes might be present in your sample, right, wherever you take them. Okay, so that's the bottom line, comparing these random DNA sequences with the reference database of antibiotic resistance genes, or ARGs, we call antibiotic resistance genes, ARGs. Okay, so the question is, why do we want to use metagenomics to study ARGs? So thanks to Dave, who has a lot of acronym already introduced, and he introduced a lot of things like novel genetic elements, so I will not explain them, but in my talk really. So why use metagenomics to study ARGs? The pros is including these main points. It's fast. You take some samples from anywhere you are interested. Okay, human gut, right? Animal gut, feces, okay, urine, whatever, and you generate sequences, and then you can annotate you can annotate them in terms of ARGs. So it's fast. It's very cheap nowadays. A few $100, you can generate a lot of sequences. And And then, of course, it provides a more complete picture of all the known ART types. So, unlike the traditional way studying ARTs, you focus on one, particular one. And with metagenomics data, you can have a more complete picture of the kinds of ARTs that might be present in your sample. So you get a more global view. The con is that it's less accurate, so because it generates random pieces of the DNAs, it could be from anywhere, right? So it might be less accurate as more targeted studies of ARTs. And the other one is more challenging. It's more of a challenge than counts. So it's big data. Usually generates our collaborators generate hundreds of gigabytes of data just from one sequencing runs. So those, you really need programming skills to handle them. So here's an example. So the previous one, you take samples from any environments you are interested in, generate those sequences, and then you can actually get through the computational pipeline, you can annotate the kind of ARTs that are present in the sample, like what I showed you here as an example. So in terms of what kind of ARTs, and in terms of the abundance in that sample, right? So this is just an example of what you can do through metagenomics analysis. Okay, so moving on to how we contribute to this research area, which is ART computational system. So these are the tools that we have developed in the past couple of years. I actually moved into this area really maybe 2 years ago. But I have a very capable student, graduate student. Basically, this is his PhD thesis. So this is really a series of a suite of ART annotation systems that he has developed so far. So I will talk about each one of these. I think for this audience, there's the expertise is quite diverse. So my mission today, I hope, is really introduce you these systems we have developed so far, and maybe help you to if you have data, you don't know where you can submit your data to, or maybe this makes you think, oh, I can actually generate metagenomic samples and use these tools to answer the questions that I'm interested in related to AMRs or ARTs. Okay, so I will move on to talk about each component of these computational tools we established. So the first one is Metastorm. It is a public resource for customizable metagenomics annotation. So this is published 2 years ago. So we have basically, this is what it does. I don't want to go into details, but all you need is just click one button. Of course, you need to register and all that. With one click of button, you will get the annotation of the things that you are interested in, such as the kind of bacteria. You submit your metagenomic data, you will get the analysis results for the kind of bacteria that are present in your sample, and if you're interested in ARTs, the kind of antibiotic resistance genes in your sample, and how abundant each one of them is. So you can submit your data through this system, get results quickly. So this is just some statistics. We have more than 20 TB of data already submitted through all over the world. So these are the heat maps, for example, the bar plot. These are different samples, for example, and these are the abundances of different ARTs in the sample. So you can make a sample comparison for ARTs. Okay. So moving on to the second tool or system we have is called Deep ART. So this one is really aiming to improve the current annotation of ARTs using deep learning approach. So the traditional way, okay, or the gold standard in the field of annotating ARTs is that you have this ART database, right? ART database, so ART 1, 2, 3, etc. Of known ARTs. And then you have all these reads or sequences you generate from the metagenomic, sequencer. Okay. So we want to compare these reads, hundreds of millions of them, to this known ART database to try to infer what kind of ARTs might be present in your sample. So this is through sequence comparison. So you want to compare this short read or short sequence against this library of database of sequences, known ARTs. And you want to align them, and then, so for example, this read can be aligned to multiple ARTs, and through this alignment, so each alignment has a score. Like, for example, how this sequence is similar to this ART here. So I will have a score, and then the gold standard is called the best hit approach. This will pick up the alignment with the highest score. Okay, and then that is the annotation of your read of this purple read here. So this is basically the main thrust or the main principle with which that you annotate these unknown reads, okay, in your metagenomic samples. Okay, so how good is this gold standard or commonly practiced, approach to annotate ARTs? So here's the study we did. We compared these two databases. So we compared CARD and ARDB. So these are two major ART databases used in the field. Gold standard, okay. Manually annotated ARTs, which people have high confidence for. And then we compare, we did a pairwise comparison and try to look at the percentage. So here is percentage identity, right? So how two sequences are identical to each other, okay? So here's a base score. So I will not go into details, but obviously the higher this score, is, the better. The more confidence your annotation is, right? You compare two strings. So the idea here is that this best hit approach or traditional approach use a strict identity cutoff. So a lot of them use 80%. So if my sequence similar to this, say, tetracycline beta-lactate makes resistance genes. So if it is 80% or above, then I consider this piece is I annotated to beta-lactams. Okay. If it is below that, then I will not consider it. Okay. So that's the idea there, strict cutoff. So you can see a lot of these, okay? So in the field, actually, there's a line here, So anything greater than this side is highly likely non-random. It's highly likely to be true, actually, ARTs. But if you use the strict cutoff, you see how many genes, a lot of them are potentially, ARTs get thrown out by the gold standard or by the common practice in the field, best hit approach. Okay, so a lot of this will result in a lot of false negatives, right? And then the other one is there's a universal cutoff of 50%. So this is a lot of things in this field is kind of artificial or you decide a cutoff. So here there's also people use 50% cutoff. So with this one, you could run into also including a lot of false positives. So these are two identity cutoffs people use. So here, okay, we did another experiment, and you can see so we take some reads, okay, so you have the ARGs, and we take some random pieces from the ARGs. So we know this piece is actually from that ARG, right? So they should be positive, two positives. But then you can see, based on the cutoff, identity cutoff, if it is using 80%, you left out a lot of two positives, right? So false negatives. You will have very high false negatives. Okay, so that's our best hit approach. That's the problem with that. So here, basically, you run into these two types errors. Type 1 error has high false positives, or have the other run into the other problem is have high false negatives. So if you use very high cutoff, you will run into this high percentage of false negatives. True ARTs, but they are not annotated as ARTs. Okay, so that's just my student favorite graph of explaining these two types of errors. So the point is, the point is that this traditional basket approach is not good enough. So we propose a solution using machine learning approach to predict ARTs by taking into account the distribution of all the hits. So if you remember my previous graph here, that when we annotate, we have this read and match to this library of ARTs, right? So we'll have some distribution of these hits. We'll have some distribution of the scores, identity scores. So instead of looking the highest or the best hit, the highest score, we want to look at the distribution of these scores. So that we introduce the deep ART system that can minimize both false positives and false negatives using a deep learning approach. Okay, so what is my time? Sorry, when did I start?
You have 10 minutes.
Oh, 10 minutes, sorry. Okay, all right. So, okay, take my word for it. Okay, this is a good system, very solid, high performance. I will not go into the details or bore you with the technical computer science stuff, but we have the paper, and this is using the deep learning approach to predict antibiotic resistance genes. Okay, so I will just move on to the.
And it's okay. 10 minutes is a long time.
Okay, yeah, I still have 50 slides. Sorry, okay. I'll keep you guys here forever, okay? So, yeah, anyway, so trust me, our system outperforms the traditional basket approach. So we compared using simulated data. We also prove it by doing using real data. So we actually have this deep ART tested on a new set of novel ARTs. So this is published by recently, last year, by Liqing Larson's group. They identified 76 novel beta lactamases ARTs. And they actually experimentally validated, it is true ART. So with our system, none of them were present in our original reference database. But then through the deep learning neural network, we were able to predict 65 with very high confidence, high probability that these sequences are actually the ARGs or beta-lactamases. So 80%, 85% of accuracies. But with the gold standard basket approach, you will not pick up them because they have very low identity to the known beta-lactamases genes. Okay. So if you want to do something with ARTs, if you generate metagenomics data or if you have long sequences, you can use our system to annotate antibiotic resistance genes through one click of button. Very simple. Okay, so this is the web server we have, and that's the user statistics already. So I will talk about the third one, which is ART miner. So I think I will talk about the big ideas. What kind of things we're using from computer science to help, to collect things or to help people analyze big data, process them, analyze them, getting results and make the results meaningful. So this is really actually an antimicrobial gene resistance gene database annotation system. So we know that identifying or discovering a new antibiotic resistance gene is very difficult, right? So traditionally you have to really test it, functional assays, right, to see whether this gene actually confers antibiotic resistance, which is very slow, slow lab work, very time-consuming, requires also expertise in domain knowledge. So curation of ARTs with high quality is very difficult. That's the problem. Because right now we're relying on the CAR database, which is highly used for antibiotic resistance gene annotation. That only contains 2,000 antibiotic resistance genes. Only 2,000. Come on, we got to have more, right? Antibiotic resistance genes, basically you will use a lot of people. So you have a lot of people here, you have this task. For example, you want to annotate, I take a picture of this room, and then I let, people on the street, okay, I give you 1 cent, annotate, just the circle, the human head in this picture, right? So that's a very simple task. Kids can do even. So they get some money reward if they circle, this is a human head, this is an animal head, something like that. So these are the tasks you gave them, and they can get monetary reward for doing that. So these are usually simple tasks, or set of simple tasks. So, okay, so crowdsourcing has been used in other fields, in biomedical field, to annotate the EMR, so electronic EHR, so electronic health records, right? So annotate those documents using the general crowd, okay, who have no expertise, who may not have any expertise. So the thing there is that we have wisdom of the crowd, okay? Collectively, we are smarter, right? Maybe individually, maybe not, but collectively, we might be able to perform something magnificent. So this is the system we have in place, implemented to garner the power of the crowd. I will not go into details. This is the user interface. It's called ART Miner. It's live online. You can actually, if you're interested in AMR, AMG annotation, you can actually contribute. So this can be potentially, broadcasted and get all the people, crowd or public involved in this, AMGs or AMRs, rather than, a set of experts, right, working on the system, which might not be sustainable or, not as powerful. So you can actually help annotation of ARGs. You don't need to have any expertise. So the point is, we did a lot of experiments. We want to compare, right? So you want to know, is general public, are they as good as experts, right? If they don't have any knowledge about AMR, how can they annotate ARGs or AMRs? So we did some experiments comparing experts versus crowd's performance. And you can see we have 3 experts with domain knowledge on ARGs or AMRs. And so, and then the general crowd, so 33 of the 35 genes labeled via crowdsourcing match the expert curation. So these numbers, and I have other results to show that the crowd is as intelligent as the expert. Okay, so that's the collective wisdom of the crowd. So this is something that you can also go to and help, contribute if you have some time to kill. Okay, like lunchtime or something. Yeah. So we have an Amazon, project there. You can get 5 cents or 2 cents for doing that, and people actually do it, and they did very well compared to the expert annotation. So it is a very effective, amazingly effective way to get, the resource from general public and bring their awareness of, this threat. Okay, so the fourth thing is really the nano ARGs. The nano ARGs is, we have this nano ARG system for annotating long nanopore sequences. So if you have long sequences, I was talking about metagenomic sequences. The sequences metagenomics data have is usually about 100 base pairs, 150 base pairs, it's short, right? So usually ARG is, 1,000 base pairs or something like that. So you only get a piece. It's the information you get from that is not reliable as if you get, really long sequences. So that's the bottom line. And that's the power of long nanopore sequence platform. So of course, the longer sequences you can generate, the more confident you have for your computational inference. So this is the first one that is built from Oxford nanotechnology. So they introduced this Minion. This is the first portable DNA sequencer. So basically, you go collect the dirt. Okay, so you collect the dirt, and then all you need is prepare going through the kits and all that, right? Prepare, extract DNAs, and then use this pipette, and then made of ETCGs on your computer. It's like that, so it's just size of your flash drive. Very, very convenient. Bring it anywhere. I go to the glacier. All of a sudden, I want to do some research. Okay, so I sample it and I can do it right there. Right, convenient, very nice, and it generates long sequences. You can see about 5 KB, 5,000 base pairs, up to the max is 100 KB. So very long. You can get long sequences. So why don't we just all jump to this, right? So there's pros and cons as with any biotechnology. It generates longer sequences, enable you to infer things more confidently. But at the same time, it has these cons, which is really, it's not here, I guess. But the thing is that it has high error rate, okay, so higher error rate. I will not talk into details about that. So what I want to say is nanopore sequencing advantages. So why do we care? Dave has a lot of slides, scaring slides about mobile genetic elements, integrons, and these cassettes and things, right, to move things around. So that means ARG does not work alone. You get metal resistance genes, you get all these things together, okay? Plasmid, they move things around, right? So these crazy stuff. So if you have only a small piece, then you can't get the entire picture, right? So if you have only partial, then you cannot get, you don't know, say you only have this small piece. You don't know whether they're linked to other ARGs, to other plasmid, or to other metal resistance genes. So you need a longer piece sequences to infer, the context of ARGs, or the neighbors, who is next to who, right? So we want long sequences, really long sequences. Ideally, if you generate the entire bacterial genome, then it's easy, right? Okay, so that's the motivation of using that's the motivation of using nanopore sequencing platform. Sorry, I had 2 cups of coffee, so I'm really like on the top or something. So there's no good systems for annotating these long sequences, and we developed nano ARG to annotate these long sequences or long reads generated from nanopore. But if you have long sequences, it doesn't have to be from nanopore. Any long sequences, we can help you to annotate. This is the architecture of the platform. You don't need to worry about. And we have high performance computing at Virginia Tech that meets all the computation, does all the computation for you. This is your interface, user interface. You just need to log in, create an account, and then you can have your projects, and you can have your samples, and you can visualize, run your results, visualize it, store it, and do all kinds of stuff. So these are the things that you can get from our system, Nano ARG. So all you need is input your sequence, right? Sequence file. And then we have these modules analyzing ARGs, analyzing metal resistance genes, analyzing mobile genetic elements, functional genes, and finally taxonomy. So what kind of bacteria are there? Yeah, 2 minutes, okay, all right. So in terms of abundance, network analysis, co-occurrence, and critically important pathogens. So we can tell you all about it, hopefully, okay? So if you have, so this is the annotation results I will not show, and this is the co-occurrence. You can see some of them are labeled plasmid. Because we are able to generate long sequences, we can annotate them beautifully like this. Okay, we don't need to put them together, because putting them together, short pieces put together, can generate errors. And this is a network, allows you to infer what ARGs tend to occur together, or occur together with metal resistance, or occur together with mobilomes, different mobilomes. And the very nice thing identified from our analysis is that from our platform is that we found this highly connected network, which actually turns out to be Alpha. This thing is related to multi-drug metal resistance. So very nice if you have long sequences, we can really do the analysis. So those are things you can do. I will not go into detail. And the last thing, okay, is meta compare. So we have another system need a system to quantify resistance risk, right? The collection of ARTs in your sample, the kind of resistance, so the kind of resistance are different, right? You sample the resistance in hospital wastewater versus you sample the resistance in, say, Arctic glacier, okay? So obviously, the latter would be much less risky than the former, right? Hospital wastewater should be very highly risky in terms of ART, in terms of antibiotic resistance. So the thing is that there's no such computational system to evaluate. A lot of current studies are generating resistance. So I take the dirt and I generate it, and I see a lot of this and a lot of that ARTs. But I don't know if I compare different samples. I don't know which one is riskier, right? I need some quantitative metric to do this. So in 2015, Martinez et al. They actually proposed a conceptual framework to evaluate the resistance risks. This is purely conceptual. So you go through this decision tree, and then at the end, the highly risk, they have alert levels. It's called response alert levels. This is the highest risk. If everything is yes, then you have this highest risk level in your sample. And 7 is the least. However, that is conceptual. It's not computational, so you can't quantify it still. And we introduced this quantification system that prioritizes the resistance risk or ranks the resistance risk. Of course, compared with the previous diagram, you can see diagram it's much more simplified, right? Because lots of things are hard to quantify. So our framework is really, our framework is really much simplified, but it allows you to identify the highly potential, highly risky samples from from the environment. So this is the pipeline. Eventually, your samples would be projected in this 3D graph, where this represents the highest risk point. And then your samples, the closer it is to this highest point, the more risky, the more riskier your sample may have, or in terms of resistance risk. So these are the things that we did the test on. And you can see the hospital this is the hospital sewage, and then the diary lagoon, and then there's wastewater treatment plant effluent. So in terms of resistance risk, highest one is indeed from hospital sewage. Second is diary lagoon. So these are the metrics we introduced. I will not talk about really. So we have a resistance risk score to evaluate your samples. Okay, so to summarize, these are things. I'm way over the time. Sorry about that. So this is really, the system that you can do a lot of things with, and it's easy. And I hope you can, it helps you in whatever ways, in terms of generating, data or generating the kind of research questions that this might inspire you. So lastly, I want to acknowledge my students. So Gustavo, I highlighted here. That's pretty much his thesis, PhD thesis, right here. Okay. So he's very, very amazing, right? If you get a really, really good PhD student, they can be like, they can be doing like 5 or 6 students or even more students work. That's what I have, okay, precious. So he did all the things, okay, in the last 2 years actually. And this is the mean quantifying the resistance risk angle. And I have other students who develop pipelines for annotating viruses in metagenomic sample, which viruses we know can also help, contribute to AMR. And these are my collaborators. Amy Cruden is the main person that, started collaboration, in this brought us into this field of, antibiotic resistance. Peter and these are from different departments. And this is the funding agency, so we are funded by NSFR. Diana is part of the team, so Peter is the PI, then we got funding last year from USDA. This is the title, so basically it's developing computational tools to identify critical control point for mitigating the spread of the antibiotic resistance in agrosystems. So the idea there is really leveraging our collaborators' data from farm to fork. So basically, you look at how manure treatment, impact the spread of ARTs all the way to the, vegetables we eat, and we want to identify the points of entry, that is critical for AMR spread. Sorry, that's a lot of time.
Yeah. Great. Again, incredibly fascinating, and fascinating as a pair of presentations to start off. So, speaking of farm to fork, we're about to move to the fork part of that very soon, and we'll invite all of you, even if you have to leave, you can take a little bit of food along with you. And we're going to invite everyone back at 3 o'clock. We're going to have a small working session with the invited guests, and then all of you who are students and so forth, certainly you